Hello guys, welcome to the channel. So as you know, we are studying about the carbohydrates, and last time we studied about the introduction to monosaccharides. So continuing to the topic, so the monosaccharide, now we'll discuss that the common monosaccharide have a cyclic structure. What does that mean? So thus as far we saw that the all aldoses and the ketoses like this right, aldoses and the ketoses uh, have represented as an straight chain molecule, right? This is a straight chain molecule, but in fact in an aqueous solution, aldotetrosis and all monosaccharides with 5 or more carbon atoms in the backbone, like this is 6, like with the 5 or more carbon atoms in the backbone, it's not actually a straight chain but occurs predominantly in a cyclic or a ring structure, in which this carbonyl group, as you can see, the carbonyl group has formed a covalent bond. It forms a covalent bond with the oxygen of a hydroxyl group. So this forms a covalent bond with the oxygen of the hydroxyl group of any along the chain of any other. So this is how it might make a ring structure. Right, so the formation of this ring structure is the result of a general reaction. How this forms? This is because of the general reaction between an alcohols and the aldehydes. So I will show you our ketones to form a derivatives called hemiacetals or hemicetals. Alright, so I'll show you here a simple formula. Alright, so this is the example for the ketones, or sorry, the aldehyde. Okay, so this is, write it down as an aldehyde. So this is the same as a ketone as well. So for example, okay, so when an aldehyde or a ketone with react with, this is an alcohol, right? So how is it is an aldehyde? So this is it's a double O, right? That's the carbonyl group, and so this R1 is this part of that replaces the R1. And for the alcohol, if you know the formula for the alcohol is COOH, so that's the hydroxyl for that, and then the R2, correct? So this is the alcohol. So when aldehyde and alcohol reacts in a 1-to-1 ratio to, that will yield a hemiacetal. So this will be hemiacetal. In the same way, if it's like ketone and alcohol, it will lead hemiacetal. Oops, I'm in the way for ketones. Hemiacetal for the ketones, right? This is if it's the ketones, right? So that will create a new chiral center at the carbonyl group again, right? So substitution, like this is just a single one, but the substitution of a second alcohol molecule produces an acetal or a ketal, right? So this will be and just an acetal, right? And when the second alcohol is a part of another sugar molecule, when this second alcohol is the part of, this is a part of another sugar molecule, the bond that is produced is called the glycosidic bond. So this is how, why I explained how the glycosidic bond is created. So right, so this is how it, so this is for the ring center. So when the aldehyde connects with the alcohol, right, it gives hemiacetal. When the second alcohol replaces again it will give acetal, and when the second alcohol is a part of any other sugar molecule, the bond produced is a glycosidic bond. What does that mean? When the two molecules that react are monosaccharide, the acetal or the kettle formed is a disaccharide. So as I mentioned, so if there is a ketone, so it will be hemikettle, and this will be just after second alcohol, it will be just kettle. All right. And when the two monosaccharides join, it is disaccharide, and the bond is glycosidic bond, as such as in covalent bond. So that was for the hemikettles or hemiacetals and the glycosidic bond. So now you will see a picture on your screen. So that is the reaction with the first molecule, as we discussed, creates an additional chiral center, right? Now, because the alcohol can add in either of the two ways, attacking either the front or the back of the carbonyl carbon, right? So it can attack the front or the back of the carbonyl carbon, as you can see on your screen, that is as highlighted, right? So the reaction can produce either of two stereoisomeric configuration denoted as an alpha and beta. So for example, D-glucose, right, exists in a solution as intramolecular hemiacetal, right? So the glucose, D-glucose, dextro, right, exists in a solution as an intramolecular hemiacetal in which free hydroxyl group at C5 has reacted with aldehydic C1, right? So as you can see in your screen, so the highlighted one, right? So the hydrox, what it does, the intermolecular hemiacetal, with that the free hydroxyl that you can see the C5, that's the carbon number 5, and the numbers are given on the carbons, has reacted with the aldehydic C1. So this is how the ring form, rendering the later carbon asymmetric and producing the two possible stereoisomer designated alpha and beta. And this is how the two stereoisomers are made. So the isomeric forms of the monosaccharide that differ only in their configuration about the hemiacetal and the hemikettle carbon atoms are called anomers, right? So what is the isomeric forms of monosaccharide that differ only in their configuration about hemiacetal or hemikettle carbon atoms, right? That's only the difference that we saw here, this one. So any monosaccharide that are differ in the configuration of hemiacetal or hemikettle carbon atoms are called anomers, and the carbonyl carbon atom, this one, is called an anomeric carbon. And on your screen you can see the another picture. So the six-membered ring compounds are called pyranoses as well. Why? Because they resemble the six-membered ring compound pyran. So the systematic names for the two rings forms of the D-glucose are therefore alpha D-glucopyranose and beta D-glucopyranose, right? So ketohexoses such as fructose also occurs as cyclic compounds. So fructose also can occur as a cyclic compound with alpha and beta anomeric forms. So how? In this compound, the hydroxyl group at C5, remember the hydroxyl group C5, or it can be C6, react with the keto group at C2. Why? Because the ketone can exist only in the second level. So that's why if it's the terminal end, this is the aldehyde, and if it's on any of this C2 to C5, it can be ketones, right? So in this compound, the hydroxyl group at C5 or C6 react with the keto group at the carbonyl group, that is at the C2, to form a furanose or pyranose, right? Ring containing a hemikettle linkage. So the D-fructose readily forms the furanose ring. Then the more common anomer of this sugar is combined to form their combine forms or in derivatives in the beta D fructofuranase. All right, so this is just a basic chemistry for monosaccharide. All right, so let me this show you something as well. Right, so this is, for example, beta D glucose, right? So the organisms contains a variety of hexose derivatives. So not only just D glucose. Hexose means that's the glucose, right? So not only glucose, but it has many other derivatives. So like in addition to the simple hexoses, that's kind of glucose, galactose, mannose, there are many other sugar derivatives in which the hydroxyl group, that is this kind of group, right? Hydroxyl group in the parent compound is replaced with another substituent, or a carbon atom is oxidized, right? Carbon atom is oxidized to a carboxyl group. Instead of carbonyl, it can oxidize to a carboxyl group. Right, so what does that mean? So for example, like this hydroxyl group, as you can see this one, if it's replaced by an amino group, I have to draw another one. Right, so this hydroxyl group, if it's replaced by the amino group, so instead of this beta D glucose, it will be now glucosamine. If it's like galactose and if it's replaced by amine group, it will be galactomine. Glucosamine or galactosamine and manosamine. So the hydroxyl group at the C2, right? It's a carbon 2, right? Of the parent compound is replaced with the amino group. It can form this glucosamine, galactosamine, right? So the amino group is commonly condensed with acetic acid. So this amino group is commonly condensed with acetic acid, as in an acetyl glucosamine. So what does that mean? So this amino group, so this amino group is actually commonly condensed with the acetic acid. So that means it is usually it's not alone, but sometimes it's with the acetic acid. So it has, right, this attachment together, and this is how become, so an acetyl beta D glucosamine, right? So this will become an acetyl beta D glucosamine. So this is the amino group is commonly, this amino group that we saw before, it's commonly condensed with an acetic acid and it forms an acetyl glucosamine. So this glucosamine derivatives is a part of many structural polymers. So what's the use of this glucosamine? It's a part of many structural polymers, including those of the bacterial cell wall. So substitution of a hydrogen for hydroxyl group at C6 of L-galactose or L-mannose, right, produces L-fructose. So L-fructose is found in complex oligosaccharides, components of glycoprotein and glycolipid. L-ramose is found in the plant polysaccharide. So these are the use of different hexose derivatives, correct? And you can see on your screen the picture of different other than the glucosamine, right? Right, so the oxidation of carbonyl carbon of glucose of carboxyl level produces gluconic acid, right? So this at the carbonyl level, what it does, it produces gluconic acid that is used in a medicine as an innocuous counterion when administering positively charged drugs such as quinine or ions such as calcium. So this is how it is used. Other aldoses yield like aldonic acid. So the oxidation of carbon at other end, that's the other end, right, of the carbon chain C6 of glucose, galactose or mannose. So the oxidation here, that forms a corresponding uronic acid. So the glucouronic, so this can be instead of glucose, it can be glucouronic, galactouronic or manouronic. So both the aldonic and the uronic acid forms a stable intramolecular esters called lactones. Right, so both the aldonic, so if it's the oxidation here, so it will produce an gluconic acid, that is the aldonic acid, and if it's oxidation here, right, so it will produce a uronic acid. So both aldonic and uronic acid form stable intramolecular esters called lactones, right? And that you can see on your screen as well. So what kind of lactones? So the sialic acid are a family of sugars with the same 9 carbon backbones. One of them is that you are seeing an acetyl neuraminic acid, that is often referred to as a sialic acid, is a derivative of an acetyl manosamine that occurs in many glycoproteins and glycolipids of animal cell surfaces, providing sites of recognition by other cells or extracellular carbohydrates binding proteins. So the carboxylic acid groups of acidic sugar derivatives are ionized at pH 7, and compounds are therefore correctly named as the carboxylates, glucuronate or galacturonate, and so forth. Right. So in the synthesis and metabolism of the carbohydrates, the intermediates are very often not sugar themselves, but there are phosphorylated derivatives, right? So when the carbohydrate is synthesized, right, or metabolism, so it's not always sugar, but it is mostly phosphorylated derivatives. Condensation of phosphoric acid with one of the hydroxyl group of the sugar forms a phosphate ester, as in the glucose 6 phosphate, as that you can see on your screen as well. So the first metabolite in the pathway by which the most organism oxidize glucose for energy, right? The glucose 6 phosphate is the first metabolite in the pathway by which the organism oxidizes glucose for energy. So the sugar phosphates are relatively stable at neutral pH and bear a negative charge. So one effect of sugar phosphorylation within the cell is to trap the sugar inside the cell, and most cells do not have the plasma membrane transporters for the phosphorylated sugar. So the phosphorylation also activates sugars for subsequent chemical transformation, and several important phosphorylated derivatives of sugars are a component of nucleotides too. Alright, so this was the introduction and the details of monosaccharides, so the derivatives of monosaccharide. Alright guys, so we'll see you in the next video. We'll continue with this. Alright, thank you guys for watching, and don't forget to subscribe.